Good, good morning, everyone. My name is Khoa Huynh. I am a PhD candidate at Virginia Commonwealth University in the Department of Biostatistics. So today I will talk about our method, the TACIT, the threshold-based assignment for cell type in spatial omic datasets. So as you may know, technology for the omic evolved very quickly. So first we have the book where we have a lot of like single cell mixed together. And then after that, we have the single cell where we're able to capture the individual cell. Uh, and then moving on later technology where we have the spatial omic data where not only we capture the single cell level, but also we capture the location of the single cell in the tissue. So here is the typical spatial workflow here. So for example, here we need to pre-processing the image, collecting the tissue, and then pre-processing the image. We do cell segmentation to capturing the cell. And then from here, we're gonna have the marker intensity. And then using this marker intensity, we're able to identify the cell type. And also like after that, we're gonna do the downstream analysis for the spatial, for example, like comparing between two different conditions or looking at like the neighborhood of the cell type. So cell type identification is very important. It affects the downstream analysis and the conclusion of the result. However, like cell type have the very challenging, for example, here is the noise from segmentation. So for example, here I show you the yellow color in the box is the one that have clear segmentation for the cell, but for the green one is like the mixture of a lot of like cells, so it's very difficult to estimate or quantify those cells in that area. And another situation is that in this figure we have the same tissue and then we run three experiment in that same tissue. So we have the single cell RNA, the spatial proteomic, and then spatial transcriptomic. In the spatial transcriptomic, here is the Muscov data. And as you can see, for the single cell RNA, it's very well separate because we have like 20,000 genes to capture only like 10 or 20 cell types. However, for the spatial proteomic, we only have like around like 56 markers to capture the same number of cell type, and also like spatial transcriptomic we have around like 300 to 5,000, but it's also like very poorly separated because like it's due to sparse, the data is very sparse. And then another difficult challenging is like the number of clustering we need to define, and also like difficult to capture the rare cell type, and we need to re-clustering every time so that we can have better clustering. So therefore, we derive the method TACIT. Here is the overview of the method. So first, we're gonna start with either like spatial transcriptomic or spatial proteomic, and then we do segmentation. And then we also need another component is the cell type recipe. This cell type recipe is the list of the cell type and the list of the signature corresponding to the signature of the gene and marker corresponding to that cell type. And of course, we need to have the cell feature matrix, and then we're gonna do clustering to capture the microcluster. So we gonna have like 100 to 200 cluster, very small cluster. In each cluster, we have around like 500 to 1,000 cells on it. And then the goal here is to try to also capture the rare cell type. Another one is after that, we're gonna have calculate the cell type relevance score, which is the matrix multiplication between the two inputs. And then from here, we're gonna derive the threshold for each of the cell type relevance score. So if the score that above the threshold, we're gonna believe that is the corresponding cell type, and if it's below the threshold, it's gonna be other cell type. So here is how we derive the threshold. So first we're gonna capture the microcluster and then rank them from low to high based on the cell type relevance score. And then from here we derive two groups. One is low relevant group and one is high relevant group. And then using that, we separate them by using the segmental regression, and then derive the threshold based on the two of the group here. And then after that, we're gonna have after we're doing that for all of the self-type relevance score, we're gonna have this matrix. So this matrix gonna represent the clean cell, which means that the cell only passing one threshold, and then another one is like the mixed cell, where is the cell that like have passing multiple thresholds. So we're gonna resolve that based on the k-niche neighbor. So as you can see here, I'm giving you one example. So the lean cell type is the red one, and the clean cell type 2 is the blue one, and the mixed cell type is the black one that's mixed between two of the cell type. And if we focus on the feature, if we're using full feature, it's very difficult to separate. But like if we use like relevant feature, like signature feature of the cell type 1 and cell type 2, it's very easy for us to separate them. So at the end, we're gonna have the final cell types based on that, and with the matrix representing the cell type, and then representing the cell type on the row, and then the column gonna be the marker. So here we benchmarking our method with the spatial proteomic data. Here for the colorectal cancer, this is the public data set, and we comparing with recent methods like Celasta and Signa, and also another one is Loven. We have higher score for the recall precision and F1, and I also showing the spatial plot, the U map and the heat map represent those four right here. And then we compare with the spatial transcriptomic, where because like Celesta only work on the proteomics, so and then for the Signa, when we run it, it have a lot of unknowns. So in here we only compare with like the Loven, and also like only the Loven one, and we also achieve the higher score comparing to the Loven. And then here is our like in-house data set for the Axiom data. So in here, we're gonna using the single cell to derive the list of signature, and then using that to run our method. So as you can see here, for the TACIT on the top, and then when we compare with Seurat transfer and then Seurat, you can see like three groups here is very clear comparing to the mixture of the group of the Seurat one in here. And then when we look at the UMAP, for example, like I'm giving you two types of the duct cell. As you can see, the TACIT able to separate them clearly, but like the Seurat transfer and then Seurat is very poorly separate them. And we also did the same thing for the subset of the T cell. Those are very difficult and challenging to identify or capture them. And then after that, after doing like model, we have the technology, like we have the way to combine both of the spatial proteomic and then spatial transcriptomic. So we're gonna using the same mask, so that we have the matching cell ID between like spatial transcriptomic and spatial proteomic. And from here, we also see that like in here is for the TOS, for the region of a lot of like immune function. And we can see like here we capture more of diversity of the immunes compared to like for the Seurat one is only like capturing all of it is just like the B cell. And we also showing the cell connection between the cell type, comparing between like Seurat and TACIT, our method here. So after doing this, we also, here's the first paper that we using the threshold, and it's published in Nature, and then we derive our method to have more, because we want to scale our data. So we cannot do manual threshold. So that's why we come up with our paper here. And then they two more paper that application of the TACIT that also in the preprint. So, and here's thank you for my collaborator in LOCI and NIX, and also my advisor, Dr. Jinji Liu. for this one.